Are at the hour and it looks like we're ready to get started. So welcome to the Knowledge Portal Network webinar series. I'm Julie Juergens. For those of you who don't know me, I'm sure you're used to my colleagues Mackenzie Brandis and Noel Burt, who normally lead this series. They unfortunately couldn't be here with us today, so I'm going to be hosting in their stead. We want this to be a way for our users to get cutting-edge access to new resources and tools that have just come online. So today we're going to be introducing a brand new resource called PanKbase with you, sharing it and getting your feedback. We have this session being recorded and we're going to post it to our YouTube channel once it's finished. So if you're unable to watch the full thing or have colleagues that wanted to join and couldn't, please direct them to the YouTube channel. If you have any questions as we go throughout, I'm going to be monitoring the chat, so feel free to post those there and we'll take questions at the end. A few announcements before we get started. As you know, this is a regular series. We have these webinars and/or focus groups every month. So coming up next will be the Mammalian Adipose Tissue Knowledge Portal and the Nephrotic Syndrome Knowledge Portal. And our next event in the series is on March 13th, same time as today's webinar. We'd also like to give notice for our sister series, the Common Fund Data Ecosystem, or CFDE public webinar series, where we're having an event on February 28th to introduce the 4D nucleome. That talk is by Peter Park. And then March 28th, we'll have a discussion from the CFDE Knowledge Center. So with that, I'm going to get into what you all came here today for. And that's a very exciting webinar to introduce PanKbase. Now, this is, as I mentioned, a brand new resource that has been developed to be a knowledge base for the pancreas and the type 1 diabetes communities. It's available through pankbase.org. We're going to be showing you all the nuts and bolts of the site as we go through, so feel free to navigate there and click with us to follow along. We're fortunate to be joined by a group of experts from across the country. These are experts in various facets of type 1 diabetes, the pancreas, islet functionality, and computational biology. We have Anna Gloin from Stanford University, who's going to be introducing the program. I'm going to be giving a demonstration of PanKbase and what it can do. Dr. Jay Liu from University of Michigan will be talking about a component of PanKbase called PankGraph, which is a knowledge graph for the human pancreas. And finally, Marcela Brasova from Vanderbilt University is going to be talking about the future of this program. So this is a very new program at the moment. We're giving this webinar early on to get your input and your feedback and to develop a community around this resource. So we're excited to get into it. So with that, I'm going to turn it over to Anna Gloin, who will give our first presentation. Anna, go ahead and take it away.
Hey. Well, good morning, everyone. Thank you for the introduction, Julie. It's an absolute pleasure to be with you this morning and to share with you an early introduction into PanKbase. And as Julie has said, I really want to stress that we're very much at the start of our journey and we're connecting with our community now because we really want to engage you on our journey. So please put questions into the chat. We'll take them at the end after you've heard from all four of us. And please, if you have questions and comments that you think of afterwards, don't hesitate to get in touch with us because we really want you to be a partner with us as we develop this resource for our community. So who and what are we? Well, we are a multi-institution, multidisciplinary team. We have expertise in platform development, type 1 diabetes, data science, computational biology, islet biology, and experts in engagement and outreach. And we've been charged by NIDDK to put together a platform that will provide our community with data, knowledge, and analytical tools of the human pancreas pancreas to facilitate research into type 1 diabetes. And we're really looking to use modern data science tools and analysis that we can make accessible to the widest community. So you'll see here on the screen some lovely mugshots of people, many of whom you probably know, who are involved in various initiatives at NIDDK and NIH in the spheres of genomics, islet biology and data science. And we're all working together as a community to put this platform together. But of course, as most of you will know, the PIs are really only a small part of the operation, and the work gets done by our amazing team. And on here, I have some faces of some of the members of our groups, many of whom are trainees with expertise across our areas of remit, working in machine learning and AI, pancreas and islet biology, experts in type 1 and type 2 diabetes, genomics, and of course, platform development. And working with this amazing group of individuals, we are collaborating to bring together this resource. So why do we need PanKbase? Well, I think it's fair to say that we are in exciting times at the moment. Our current data ecosystem is very diverse and it is growing. We have never had so much data. And if you look at the schematic, you can see here some, not all, but of the data generation and repository efforts that are led by many colleagues across the international space. If we look at those in the middle, these are the closest sort of proximity to generating data on the human pancreas. But you can see here our ecosystem is very large and encompasses other efforts that are around genetics of diabetes, looking at genetics of other diseases, and annotation of tissues around the body. So all of these are really important, valuable resources that are generating absolutely vital data. But it's really challenging to navigate this system, to know where to find data and also how to integrate it. So I think what we're coming to you with is challenges and opportunities, because with this amazing data ecosystem that we're part of, we have never had so many challenges in trying to use it, or as many opportunities in terms of making the most of the investments that have been made by funders and colleagues. So some of the challenges that we face as a community is finding and accessing all of the data that's relevant to us, answer and address our research questions. We have challenges with a lack of standardization across these different projects, which can hinder analysis and metadata integration. There are difficulties with interoperability between resources because of the way that we've set things up, and this limits our ability to integrate different types of data across these important resources. As we get excited about AI, we're also recognizing that much of the data that we've generated over the last 10 years or so is not AI ready and ready to be fully integrated. And this is a barrier to being able to maximize investments that have been made in data generation. There's also a wealth of literature out there that is in publications. Much of it, of course, is in unstructured text, and it's more complicated to bring that into our analysis. So how do we find ways of making the most of literature and knowledge from our community? But thinking about those opportunities, we really are spoilt with huge investments that have been made by funders across the globe in generating really high quality data sets that are relevant for addressing and answering questions in the diabetes space. And we've got opportunities if we can bring this data together to gain statistical power through integration that will allow us to answer questions that have been ahead of us for some time because we haven't had power to resolve them. We've also now got opportunities for multimodal data integration, things that haven't been possible before. We can integrate different types of data from different resources. And of course, there's this wonderful opportunity now of machine learning and AI-based approaches that give us opportunities to address things that we haven't been able to do before. And I think what we're also very cognizant of is that in the field of pancreas and islet biology, we're so fortunate to be donated tissue from families and individuals that we really have a duty to ensure that every human islet that has been offered for research matters, and that we're able to get as most out of the resources that have been generated on that precious sample as possible. So this is why we think PanKbase is needed. We have this diverse and expanding data landscape, and what we really need is efforts for expert curation of the data, data harmonization, and standardization of analysis. And if we can achieve this, we can offer democratization of access to the resources by putting together a catalog of highly curated data, which has rich metadata accompanying it. We can provide a platform for biologists, for clinicians and data scientists. So a platform that works for you no matter what your entry level is of familiarity with the data. We want to offer analytical tools that can be run in any computing environment so that those of us who don't have access to supercomputers can still make the most of the data that is available. We want to work with the community to come up with robust standards for human pancreas research so that we can all work together to use data in a harmonized manner. And critically, we want to engage with experts and our community so that we can ensure that what we're doing is valuable to as many people as possible and brings in as many experts and diverse data types as possible. So as I said at the beginning, I really want to focus on the fact that we are at the very start of our journey. We have only been working together as a team for a year. So we're right here at the beginning of what is a challenging journey to make it up this mountain. And we're going to tell you what we've been doing over the last year. But really, one of the biggest take-home messages is that we want you to come on this journey with us. And we would love to hear from you so that we can bring in data sets that you're interested in working with us on, and ideas that you have for how this resource could be more useful to you along the way. So I'm going to start off with some of the examples of the work that we've done over the last year, really to highlight why we need PanKbase, because perhaps you haven't thought about some of the challenges yet that you might face when you start to use data on the human pancreas. So I'm going to start with some of the work that we've been doing across our consortium on cataloguing human islet data and providing a highly curated set of data with rich metadata. So one of the challenges that you face when you start to ask questions with human islet data and you want to move between resources, that each of these amazing efforts that are run by colleagues across the US and beyond, in Canada with Patrick McDonald and the Alberta Diabetes Institute, with the Human Pancreas Analysis Program with Klaus Kastner and colleagues, and the Integrated Islet Distribution Program with Joyce Nyland and Carmela Evans-Molina. You can see all of these valuable resources are amazing in their own right, but it's not necessarily easy to work and integrate them across studies. So you will also notice here that each of them have very different numbers of islets that and donors that are provided. The ADI dataset has just shy of 550 donors at the moment. The HPAT resource, which has very deep phenotyping, has around 170. But perhaps surprising to many is the sheer number of islets that have been distributed by the IIDP, which are just over 1,500. There are also other really valuable resources such as NPOD that provides tissue for type 1 diabetes, and they have around 600 donors. And of course, Prodo that provides islets as a commercial entity for research. So how can we bring together standards that will allow us to bring these data types together for shared analysis? Well, some of the challenges that we face is that we have to work and make sure that we're not counting one donor more than once. And this is difficult because there's been inconsistent use of RRIDs. And also there's different types and levels of clinical data available that can be a hinder to certain analysis. If we get down to the biosamples and details of islet isolation, we've got different levels of information captured by different resources on islet isolation, quality, and culturing. And then when we think about the assays and data that have been generated, we may have different depths of phenotyping and inconsistent reporting of experimental protocols. So all of these things can make it very challenging to work across all of these amazing resources and bring the data together, and can be a barrier to making the most of every islet that's out there. So our aim has been over the last year to come up with ways of capturing all human islet donors and to be able to link them to comprehensive metadata. So to do this, we've really tried to focus on a practical way of moving forward that allows us to be very transparent so that we can bring every human islet donor in, but really be transparent about the level of information that is available on that human islet donor. And to do this, we've come up with tiers of required information, desired information, and optimal information so that you as a user can make decisions on how stringent you want to be with the data that you include in your analysis. And we've really been influenced by the Heart and Powers standards that were published in 2018, which form the basis of the requirement for diabetes and diabetologia for reporting standards for using human islets in your research. We're now working very aggressively with our partners across the human islet space to encourage adoption and reporting of these resource research identifiers so that we can make it easier moving forward to link data to metadata. So some of the things that we've been doing is assembling information on the human islet donors, bringing together as much information, things like age, reported gender, BMI, diabetes status, duration of diabetes, and important additional parameters such as HLA typing and autoantibody status. We've also been assembling data on the biosamples to do with the islet isolation, and also putting together more information on the downstream experiments that have been generated on these valuable islets to do with library preparation and sequencing parameters. So what can you do once you start to bring together such a well-curated resource that's stretching across multiple islet providers? Well, we can start to think about how we would combine the data to generate single cell maps, to look at islet function, and to generate islet EQTLs. And a lot of this data, for example, has already been put together for the Human Pancreas Analysis Program, and that's on over 100 donors, and you can find beautiful data ready to use, freely available on PanKdB. But if you want to bring in over 1,000 additional donors from the IIDP, you need to be able to combine those resources. And over time, we want to be able to take this out to Patrick McDonald's data and to collaborate with him and his colleagues so that we can extend it to a further 500 donors. And then, of course, there are other donors out there and we would like to bring them into the fold. So at this point over the last year, we've really focused on integrating the Human Pancreas Analysis Program, working with our colleagues at HPAP to integrate data with IIDP donors. So once you have brought those data sets together, the sort of questions that you can answer using the knowledge derived from the integrated resource is that we can ask what cell type gene expression looks like. So what does an alpha cell look like, a beta cell? And of course, a lot of the rarer cell populations, which start to be seen in greater numbers. So we have more clarity on what genes are expressed in those rare cell types. We can ask whether or not there are changes in gene expression that are associated with a particular phenotype. We can also look at whether or not there are cell type marker genes, and of course look for co-localized EQTLs. So are there expression EQTLs in islet cell types that are associated with diabetes risk? And of course, there are many more applications that can be done once you start to derive knowledge from these combined resources. So I want to highlight a couple of the ways that we've been able to provide added confidence in our ability to link donor metadata to genotype and genomic data that has been generated on these resources. So one of the challenges that you may face if you go and download data from GEO, for example, is you'll have a set of islet samples, but you may not necessarily be able to link that back to the metadata that's been deposited and made available in IIDP because there may be insufficient information in the manuscript that has been reported. So one of the things that we've been doing is to work with our colleagues at IIDP to take genotyping on all of those samples and to be able to link it to the data that's been deposited in the publicly available databases such as GEO, so that we can link the metadata from IIDP to the data that's available in the repository. And we can do that by pulling out the genotypes from the donors in IIDP and then pulling the genotypes from the deposited data and matching to give us an idea whether or not donors are accurately aligned with their metadata. We can also rescue data, samples where we've got insufficient data so that they can be aligned with the correct donor information. And we can also, in some cases, resolve sample swaps, which we find in about 5% of the time. So this gives end users real added confidence that the data that they're using for their analysis is correctly aligned with metadata and allows us to maximize sample size. So over the last year, we've focused on two areas that our community told us that they really cared about, which was putting together a standardized framework for analyzing bulk and single cell level sequencing data from human islets. And working across our team, we've come up with a standardized pipeline that we've also containerized so that other people will be able to follow it and use it for their own data analysis. And we've put together combined matrix counts and single cell maps that are available in PanKbase, and you'll get a view of those very shortly. So to give you a sense of the numbers, we now have data on bulk RNA for just shy of 500 samples. And you can see here that these are from IIDP and from HPAP. And we've also been able to put together a single cell map from HPAP, IIDP and Proto using this carefully curated data that we've worked very hard to ensure that we've got accurate metadata reporting. And this is fantastic because what we've been able to do is build on the work of our colleagues in HPAP who've done a fantastic job working with 75 donors across 87 samples and giving us just shy of 200,000 cells. But by bringing in data from our community that's been published and released in GEO, we've been able to bring in data from IDP and Proto that gives us just over 500,000 cells now. So you can see a really fantastic resolution single cell map of the human pancreas. So my final point from this introduction in terms of why you need PanKbase and what we're looking to do is I'm just going to highlight some of the efforts that we've already made to focus on delivering robust standards for human pancreas research. And you can find this information on the PanKbase website. You can find the extensive documentation that we've put together of our processing and standards that we've used to create the resources that you hear about today. There's a summary of the data and also the QC that we've got of the resource we've currently got on offer. And also you can see our detailed workflows that we've provided in containers for anyone to run. So you can look and find details for donor metadata, data processing and QC, and also details of the data summary. So we really hope that we're being transparent and making it easy for people to see exactly what we've done and to replicate it with their own data sets. So as Julia said, we're going to move on now, and what you can expect from the rest of the session is that you're going to actually get a chance to see what PanKbase looks like, and Julie will be presenting this. And then we're going to hear about PankGraph from Jay, and then Marcella's going to give us a sense of where we're going in the future on our journey on this mountain and how you can join us. Thank you.
Fantastic. Thank you, Anna. For anybody who has questions, please feel free to pop those in the chat. We're going to take questions in the end, since some of those might be addressed as we go along. But I'm just going to jump into the next part of our session. So you've heard from Anna a really nice introduction of what PanKbase is, right? Who we are, what we are, and why we need this resource. What I'm going to be talking about is our actual initial offering. So as Anna said, we're just into the first year of this project, and this is a nascent resource that we're developing out. So we want to tell you what we've developed so far to get your feedback and think about where we can go next. Here are the major features that we're currently offering. I'm going to walk you through those one by one. First, we have a landing base for the human pancreas. We have multi-site donor metadata that's been standardized and put into one place. We have single-cell RNA-seq data that's acquired from multiple sites that's been processed and analyzed, harmonized in a standardized way, and put again into a single place. And finally, a knowledge graph for EQTLs in the human pancreas. So as we go through the website, you can follow along with me. Please navigate to pankbase.org, and you can see each of these site components in action as we go along. So if you go to pankbase.org, you'll be taken to our landing page. So this is a landing base for the human pancreas. Scrolling down will take you to our resources. So here you can learn what is actually unique to PanKbase. And this includes both of our current resources, including PankGraph, integrated maps of pancreatic islets, and donor summaries, as well as what resources are coming soon. So these are things like genetic associations and image browsers that we're in the process of developing, but don't have online yet. Stay tuned for those. If you scroll down further, you'll see various tools and workflows. These are the analyses that you can do on PankBase. So again, we're developing these out as we go along, but you can see currently our cell browser and how to find relationships among various entities like EQTLs. You can also browse examples. So these are use cases that explore what is in PankBase. We're developing these as well, but over time, these are some of the use cases that we expect that users can access through our resource. Further down on the page, you'll see our external resources. Now, as Anna mentioned, there are many existing resources in the human pancreas community that have put together extensive data, metadata, and biosamples for human pancreatic islet donors. These are precious samples that are hard to come by. And these programs have really done an amazing job of putting this information together. So what we've done is worked with these programs to provide a curated foundational knowledge page for each, where you can get more information about what each of these programs offers, how to access their websites, and the teams that develop them. Scrolling down further, you can learn about our partners like the Human Islet Research Network and many others shown here, how to collaborate with PankBase, ongoing news from us and our partners, and more. So now I'm going to talk about the donor metadata. As Anna said, this has been standardized and put into one place on PankBase, and you can access it here from our donor summary on the homepage. That takes you to this donor summary tool, which is actually an interactive app developed by our colleague Liza Bruceman and Kyle Galton's lab at UCSD. And what Liza has done is enabled us to look at the donor metadata from each of the donors. There are 3,500 in total acquired across various programs. You can filter by program just to get the information from that specific program. And importantly, you can filter by various other metadata fields. So for instance, if you want to see information about all of the donors of various diabetes statuses, you can filter in that way here. You can also access various other visualizations like these ridgeline plots, heat maps, correlation matrices, scatter plots, and PCAs, as well as views of assays by donors. So here you can see which assays have been generated for individual donors across programs. We've begun this with one of the programs, the Human Pancreas Analysis Program, or HPAP, and we plan to build this out over time to give users a snapshot of what data are available across donors. So you can see all donors that have, say, single-cell RNA-seq and single-nucleus ATAC-seq. That's our vision over time. On PankBase, you can also access donor data. And from what Anna shared previously, we know that this data has been really extensively and carefully documented and organized so that you can get to it in a centralized place. So this includes data acquired again across those external programs that worked hard to develop them. You can access that through our our data library through this resources tab up at the top. And here you can see summaries of the 3,500 donors, biosamples, assays, and other fields that you see here. If you click on the donors tab, you can get a summary of each individual donor, which program collected them, and then you can click on that donor's page to see a summary of their metadata, their RIDs, and then other information about them. You can also get a sense for what data are available. So again, we've done this just for a subset of individuals, but over time we're building this out. Clicking on the Biosamples tab will take you to a list of the 400 biosamples that have been obtained from pancreatic donors. And you can get a similar sort of view of each through this catalog here. Finally, we have lists of process results, analysis results, and workflows. And you can explore each of these on this site. If you go to the analysis results in the middle here, you'll come to a page that looks like this. And this will tell you how we did our two major analyses for the data that are currently available through our integrated cell browser that I'm going to get into next. These include single nucleus attack-seq data and RNA-seq data, both and single cell RNA-seq. So you can learn more about those data and access them here. All right, so now I'm going to jump into the integrated cell browser. This was developed by a team from multiple sites, Alex Schillen, Peril Kardutar, and Ha Vu. And you can access it here through the homepage. So the integrated cell browser currently has 3 major data sets. 2 of these are single cell RNA-seq and 1 is single nucleus attack-seq. And I want to draw your attention to this one here. This is a data set that currently has over 500,000 cells derived from the human pancreas. These were acquired from 3 programs, HPAP, IIDP, and Proto. And clicking on that will show you an integrated map of the data from all of those programs. So you can see UMaps that show cell composition, gene expression, proportions of cells per cell type, and violin plots to visualize the gene expression. Importantly, you can also filter these interactively. So say you want to look at diabetes status for this type of a graph. You can click that and it will show you the same graph broken down by diabetes status. So this is a really nice feature. You can also look at marker genes to see what the top markers are for various types of cells in the human pancreas. Finally, you can download the data using this button here on the right. As I mentioned, these data were collected and put together by many other external programs. So we want to direct you to our policies page, which you can access here at the top menu bar. And this will take you to the policies for each individual program that went into generating the primary data used to generate these collective maps. So please refer to their citation policies as their resources were integral for building these maps. All right, finally, we'll take you to the analytical library. So this is a feature that we're currently developing and that will be coming soon. But our vision for this is to provide sort of an access point for multiple different types of users to come into this PanKbase site and to perform analyses with variable levels of customization. So for someone who's an expert, they might be able to access a cloud-based workspace and customizable code. For somebody who's newer to the field, they might be able to use these custom web interfaces that let you look at the data out of the box. All right, so now we're going to talk about where you can learn more. So under the About tab here, you can see various static pages with more information about our program and some of where you can go for more information. So clicking on this project page will take you to a summary about our project. our mission, what resources we offer, more about our team, and then where we're headed. And shout out to Amanda Huber for her work on this. And finally, you can access metadata and data standards under the help menu up here. This will take you to the tools and pipelines that were used to process the PanKbase data. And we've put these here for rigor and reproducibility purposes. So these are accessible through sort of static PDFs as well as through GitHub repositories. So I know this was a lot, but as I said, we want to hear from you. So if you have any questions, comments, ideas for collaboration, please reach out to us at help@pankbase.org, and we'd be happy to show you more. So with that, I'm going to turn it over to my colleague, Jay Liu, who's going to talk about an important part of PanKbase that I did not mention yet, called PankGraph. And he's going to delve into this in great detail. So Jay, take it away.
Oh, thank you, Julie. So next, I will talk about this PanKgraph. So first, I will show you how a PanKgraph look like. So essentially, we have a knowledge graph connecting so many resources, donors, about medical entities in this knowledge base. So essentially, we use a graph to do that. So here, I only show you part of the graph, only containing 5 nodes and 5 edges, describing a genetic variant. Is a GWAS association with T1D. It's also a eQTL of a gene that is expressed in pancreas, and this gene belongs to this ontology. So here we have nodes, like a gene, and for every nodes and edges, we can have property or attributes. And then we have edges or relation that represent the knowledge. And on the edges, we can also link back to original single-cell RNA-seq data, capture this knowledge, this gene expressed in the pancreas tissue. And we also have other knowledge like whether this variant is a GWAS signal that is fine mapped causal variant. So why we need a knowledge graph in PanKbase? The first reason is a knowledge graph is more intuitive, presenting the biomedical knowledge. So just use the same example of variant that is both a GWAS signal and also eQTL signal. This is a better representation than original tabular format. We have the information in the original different tables. On the left, by using the knowledge graph, is more intuitive and also easier for computer to understand the relationship among them. So the second reason is really we have this broader vision to link different resources and provide the provenance of the knowledge. As the years goes by, we will link more and more resources, but you can imagine for some the entity like a gene CFTR, we can have different resources of knowledge about the same gene. Like in example, we have some information in PubMed, we can have another article about CFTR. In GTEx, we can have expression level about the gene, and then in string and gene ontology, they all provide some information, but they are all separated. But with our knowledge graph, we are able to identify them and integrate them with the explicit graph. So the scope of our knowledge graph, at this moment, we have 500,000 nodes, 1.6 million edges, and still growing. This is just a start. We are planning to integrate basic knowledge in T1D research and islet biology. At this moment, we have high quality data and knowledge contributed by the team and broad community. So far, we only have GTEx, a GWAS signal, eQTL signal, and fine mapping and colocalization. We will have more in the future years. We will integrate research articles about T1D. Right now, we have already over 1,000 papers extracted from HERM publications. We are planning to integrate more from PubMed. We also integrate annotations from different repositories like GTEx, Ontology, and DBSNPs, and etc. And we are also linking resources within and outside PanKbase, like entities in PanKbase data library, like donors, and also pipelines in PanKbase analytical library. For example, if this eQTL is analyzed identify from some pipeline, we will also trace back to the original pipeline generating the EQTLs. We will also link to eternal resources, like example and PubMed. We also follow this 5 principle. We want to make sure all the knowledge and data are findable, accessible, interoperable, and reusable. For doing that, we carefully identify the identities for every node. We will have the unique IDs identifier for them, like genes, variant, and disease. They all follow some identifiers. We also have schema or metadata defined. For example, for a gene, we need to have the strand information defined. We also have edges defined with according to some metadata. For example, the functional annotation need to be start from a gene to a ontology term to make sure this follows this edge. And in the end, for all these scientific concepts, we also refer to existing ontology to make sure this is well defined. By using identity, schema, and ontology, we make sure all the edges nodes follow this 5 principle. Another concept is this open knowledge graph concept. We want to make sure the pipeline we use to build PanGraph can be shared with everyone. So that is really to empower other people to build similar knowledge graph using the same set of vocabulary. For example, if DKNet want to build a similar knowledge graph, they can refer to our pipeline and using the same set of vocabulary. The power is really in the future, we are able to connect all these different knowledge graph using the same set of vocabulary. This definitely will promote the interoperation among KGs. So next, I encourage everyone to visit the website. You can either go to PanKbase and click the PanGraph tab, or directly go to pangraph.org. So this is the landing page. Right now, you can click the questions, and through the questions, that will trigger these triplets. And from the triplets, you can type in the gene or the entity of interest. In this situation, I want to identify the QTLs of the gene CFTR. After I type in this gene and click the search, that will go to the intermediate page. So that will ask you, this is the question you ask, which SNP serve as the QTL of the gene CFTR? In our knowledge base, we have 4 types of QTL result. For this particular gene CFTR, we have the pancreatic splicing QTL identified from a GTAC study. So there are 3 sets of credible sets, and then they are listed here. We also provide information about purity, lead SMP, PIP scores, and you can click to see more information. So if you click the first one, that will trigger to a result page. For this particular SNP, how does this SNP influence the splicing of CFTR? So this is a result page. We have multiple sections. We have an AI answer section like this. On the right-hand side, there's also a graph viewer that really show you how the knowledge graph connects CFTR and other entities like the SNP of your interest. I think the most exciting part is this AI answer. So I will go through this detail. For your question, the AI answer generates the following answer. So what's cool about this part is it will use LLM to generate the overview to this answer. And when we generate the answer, it will always refer to a real PubMed ID. So this is really because we have extracted knowledge from the PubMed articles. We are able to provide the provenance when we generate the answer. So this will ground the LLM and support its explainable AI. We also link the resources within and outside the PanKbase. For example, the EQTL study, the result I showed you come from the pipeline within PanKbase. We will link to the original pipeline in PanKbase analytic library. And the last part is you almost may also ask a question. This is really along the knowledge graph passes, we provide recommendations. So this is a really cool part. In the future, we can generate hypotheses based on knowledge graph and AI. We also have the API ready for everyone to access. So you can think about the website is only for you to query one at a time. But if you have the situation you want to query systematically, you have hundreds of queries, you want to do it together within your code, this is really the place you can call the API. In this situation, I show you an example how to identify all the variants linked to the gene in one signaling pathway. So you can write the cipher language like this. And by executing this query, you can return the 194 variant about 3 genes. So really to speed up and call the visit the query the knowledge graph systematically. With that, I only gave you a kind of taste of knowledge graph. Definitely it's growing. And I think it's really having the data in a format that is not just interpretable, easier for human, but also friendly towards computers. We suspect in the future we can engage a lot of AI models on our knowledge graph. We can empower scientific discoveries. I already showed you the question answering part and a little bit of hypergeneration. I think more is coming. So with that, I will pass to my colleague Marcella to talk about the future. Thank you.
Thank you, Jay. So let me share my screen. Okay, here are the slides. And oops. All right. So as you nicely heard from Anna, Julie, and Jay, this is a very young program, and I have a logo O'Hearn. It's nested. This new initiative is nested within Human Allied Research Network. Our overarching goal is to promote open science, reproducible research, data-driven scientific discoveries, especially now with the dawn of AI and machine learning technologies that was marked by 2 Nobel Prizes awarded last year. And this is really important to accelerate the translation of basic science discoveries to clinical clinical research and patient care. Let's see. Sorry. My window is blocking the slides. Okay. Sorry for that. And oops. Okay, here we go. Sorry. We hope that PanKbase will become a destination for diverse research communities, including biologists, computational scientists, and clinicians. And it will provide first-in-class education and community outreach to the general public and organ procurement organizations, which are really critical in supporting diabetes research. We are very grateful to organ donors and their families, without whom none of this would have been possible. And so everything starts with metadata, as you nicely heard from Anna, and aggregation of multimodal data sets across age, sex, disease states to provide new insights into type 1 diabetes. And really being able to incorporate cohorts covering a broad range of type 1 diabetes development, starting with individuals without diabetes as control, those who have autoantibody positivity, dysglycemia, and symptomatic type 1 diabetes. Inclusion of broad age ranges is becoming more and more important with the emergence of type 1 diabetes endotypes, and we currently do not understand the mechanisms of their development. And so Also, what is really important to think about is that emergence of autoantibodies is marked by early postnatal period, as was very nicely demonstrated by Dr. Anna Ziegler and her colleagues, as a very vulnerable period of life. So, for example, children who are 5 years, who develop autoantibodies in this first stage within first 5 years of life, have 50 to 60% chance of developing type 1 diabetes by age of 10. So, again, through survey of community, as Anna nicely mentioned, our platform started integrating first genetic risk and variants into platform setup, and also transcriptomic and epigenomic profiling. But as time goes on, we hope to incorporate data sets including tissue, overall pancreas architecture, imaging data sets, and also functional data reflecting islet physiology. And what is really important is to bridge basic science discoveries to clinical and translational research, and we are very fortunate to be supported through Office of Data Science Strategy to start developing common data elements in collaboration with our partners at University of Southern Florida. And so our goal is to integrate, to build knowledge leading to type 1 diabetes prevention. As you know, type 1 diabetes is a complex disease with a strong HLA component. And as work of Dr. Anna Ziegler showed recently, children who are born with high-risk HLA alleles have dysregulation, or rather excursions in preprandial and postprandial glucose levels prior to seroconversion. So thinking about not only genome, which is invariant, but how environment and age and sex influence other layers of information that we are trying to integrate will be very important for us to consider going forward. And this will lead to better understanding of etiology and pathophysiology of type 1 diabetes, development of diagnostic tools and therapies for those with type 1 diabetes, and defining causality that will allow us to bring in prediction modeling, and that will pave the way to type 1 diabetes prevention. This cannot happen in isolation. We are very fortunate to have a number of partners in this large data ecosystem that Anna described early on. We are building partnerships with a human pancreas analysis program that is nested also within the HERN Human Islet Research Network, and also programs such as Integrated Islet Distribution Program, Network for Pancreas Organ Donors with Diabetes, and Alberta Diabetes Institute. So working together is really critical for us to advance this initiative and path forward. We are also building partnerships with Human Cell Atlas, for example, with their integration team, focusing primarily on transcriptomics data sets. And so, as Anna said very early on, we are at the very beginning on our journey. That's a journey to the top of this mountain. Climbing mountains is really hard, but we do it because it's hard, right? And we want you to join us on this journey to the mountaintop. There are many ways you can get involved. We are assembling community board because we want to have a good pulse on what community needs. Way to contribute data is one way to get involved. Also participating in tool development. And we hope that in the future that this will be supported through opportunity funding. And please reach out to us through this email, help@pankbase.org. We want to really hear from you. Here's our QR code. please complete the survey and let us know what you think. I want to emphasize that the platform that Julie walked you through and Jay introduced, the Knowledge Graph, this is really our beta version. It was launched at 10th anniversary of Human Allied Research Network. It's a very young program, and we hope to go through full launch at sessions of American Diabetes Association in Chicago in June of this year. So please send your feedback and let us know what you think. Thank you all for your kind attention, and I will turn it over to Julie if there are any questions. Thank you.
Fantastic. Thank you to Marcella, Jay, and Anna for their fantastic presentations. And any questions from the audience? All right, so I have a question. So if users are reaching out to us, I guess what are there specific types of data that we're especially interested in integrating in at this time? And any, I don't know, any guidelines around that?
I think what I would encourage is if you think you have data that would be worth contributing or you're interested in, reach out to us. I think we want to hear. There may be things we haven't considered yet that would be really valuable. So we would rather hear from you than not hear from you. And also a lot of this is we're kind of working on it as we go. We don't necessarily have all the answers yet. So we really want this to be a partnership. So no question is too small or too silly. There are no silly questions. Just get in touch with us. This has to work for the whole community. This has to be valuable to people. So we need to know what people are interested in doing, what they want. So please, please, please email us. Lots of us. We're all very friendly. We've got. We'll make time to talk. We can Zoom you. We can do things by email. Just reach out.
Fantastic. Any other questions from the audience? If not, I think we're going to go ahead and close for today. But thank you so much to all of our fantastic panelists. Again, visit us at pankbase.org and reach out at help@pankbase.org if you want to collaborate or have any questions after the webinar. Thank you so much and hope you all have a wonderful day.